These are your chapter 5 notes. They're actually two parts. I broke this up because it's a little bit longer of a video. This is over the structure and function of large biological molecules. And we're going to be talking about carbohydrates, lipids, proteins, and nucleic acids. So we're going to kind of split it up a little bit. So carbohydrates, proteins, nucleic acids, and lipids. Lipids technically are not large enough to be true macromolecules, but they're important enough in cells that we need to talk about them, particularly important in the cell membrane. Macromolecules may also be called polymers, so using those two terms interchangeably. Long molecules made of many similar or identical building blocks, made up of monomers, repeating units that serve as the building blocks of a polymer. So mono means one, and then poly means many, and that's where you get those terms from. So monomers are the building blocks that create these larger molecules called polymers. Synthesis and breakdown of polymers. Building or breaking polymers occurs with the help of enzymes. Enzymes we're going to be talking about in chapter 8, and they are going to speed up our chemical reactions. They're going to make them happen faster. Dehydration reaction is going to be what builds polymers, and a hydrolysis reaction is going to be what breaks down a polymer back into those monomers. You do need to know the difference between these two types of reactions. The dehydration reaction, you're going to take two smaller units, okay, and you're going to link them together with a dehydration reaction. It's going to take the hydrogen end of one molecule and the hydroxide end of the other molecule. It's going to remove a water from there, and it's going to put those pieces together to kind of put them together like a puzzle. A hydrolysis reaction is going to have the opposite effect. It's actually going to break down a polymer. So you're going to need to add that water back in, add the hydrogen to one end and the hydroxide group to the other, or hydroxyl group, sorry. And then you now have two pieces. Okay, so here in a dehydration, I try to remember this because I think it's dehydrating the reaction. It's taking water from it. If we dehydrate you, we take water from you. Your body is going to seep water out. And so we're dehydrating it, we're removing water, and that's going to put the pieces together. Okay, and then a hydrolysis reaction, hydro meaning water, we're adding the water back in, and that's going to break something down into smaller pieces. Make sure you know the difference in those two. Dehydration reactions are used to build polymers from the monomers. It creates a water in the process. An example is during the formation of proteins from simple amino acids. And again, showing you this, the dehydration reaction, you're going to have one amino acid here and one amino acid here. And then you're going to remove a water, the hydrogen and the OH groups, and then now you have a larger polymer. So this might be amino acid 1, and amino acid 2, and amino acid 3, and amino acid 4. So each time you add another amino acid to that chain, you're going to remove a water, you're going to dehydrate it. Hydrolysis is disassembling the monomers where the bond between the monomers is broken with the addition of water. The H from the water attaches to one monomer, and the OH, or the hydroxyl group from the water, attaches to the other monomer. An example here is digestion, when complex polymers are broken into smaller monomers to be absorbed into the bloodstream. So you're eating a big bunch of pasta. Those are really, really complex carbohydrates. They're large. And what's going to happen in that digestion process is it's going to split each of those, split that large polymer, that carbohydrate, into simple, tiny sugars, which is why your blood glucose goes up when you eat a lot of pasta, is because you're breaking that large carbohydrate down into simple glucose sugars. And in that process, we have to add water to it. So our first category is going to be the carbohydrates. Monosaccharides are the simple sugars. Those are the building blocks. Disaccharides are double sugars, and polysaccharides are going to be many linked smaller sugars. Most sugars names end in ose, but not all. So sucrose, glucose, galactose, fructose, but not all, but that is a common ending. Major source of fuel for the cell and needed for cellular respiration. Glucose, again, is going to be the main one we use in cellular respiration. The idea here is carbohydrates are a fuel for the cell. They really fuel the cell. Sorry, I had to take a drink real quick. Some polysaccharides serve as a storage material, starch in plants and glycogen in animals, or building material for structures in the cell. Cellulose or keton. That's kind of weird spelling for it, but it's pronounced keton. Stored fuel cannot sustain an animal for long and are depleted within about a day and need to be replenished. Again, that's why you have to eat a lot. There's an issue with low carb diets. A lot of people that are on low carb diets will see a weight loss because they're not building up the sugar, because sugar, if not burned, is going to turn into fat. But a lot of times they experience a lack of energy, and that's because you really do need a constant source of carbohydrates to be your main source of energy. You just don't want to eat more than you can burn. I do that a lot, unfortunately. Cellulose is grouped uniquely, and few organisms have enzymes that can digest it, or in other words, break it down. Cows are an example of one that can. They have an extra stomach that is specialized in breaking down cellulose, which is why they can eat grass and we can't. Keton is used by arthropods to build the exoskeletons, and some fungi have it too. And here's just showing some of these sugars. Remember we talked about the carbon, the branching, and the long chains. There are also some sugars that are rings. Here's an example of an abbreviated ring structure, or here's another ring structure. Here's more. This shows that dehydration reaction here, where you're taking away this water, and you're taking 2 glucose molecules to bring about maltose, and you're taking the water away from that reaction. And this is a glucose and a fructose will produce a sucrose. And again, you're taking that water away during a dehydration reaction. And this just shows some simple structures of starches or glycogen. These are storage carbohydrates. And then cellulose, the plant cell walls are made of really highly of cellulose. And this shows you what those would look like. Very complex carbohydrates. And keton as well. This is interesting. It's actually used to make a strong and flexible surgical thread that decomposes after the wound or incision heals. So they can use that as an internal thread that they don't have to remove later. Lipids, again, remember these are not true polymers. The major function is energy storage. Lipids are grouped together because they mix poorly with water. The water molecules hydrogen bond to each other and exclude the fats. The fats consist of a glycerol and a fatty acid, also called a triglycerol or a triglyceride. There's some different types of fats here. You may have heard of these in maybe a nutrition class or health class. Saturated fats are straight. They have no double bonds. Most of these are animal fats. They are solid at room temperature. Think about when you fry up some bacon and you let it sit. That bacon grease solidifies. Omega-3 fatty acids are essential and need to be supplied in the diet. You can get those usually in fish. Although I did read an article recently about how tilapia nowadays, it's so common that it's often created on fishing farms. Like, it's not fished naturally in the wild. They have farms for it. And because of the way that they farm them, they actually really don't have the omega-3 fatty acids. So there's really no benefit to eating them anymore. Interesting. Unsaturated fats contain a double bond that bends the structure. Most plant and fish fats. Liquid at room temperature. The bend means that they can't pack closely enough to be a solid at room temperature. So they are liquid. So think about your olive oil and your vegetable oils. Trans fats, special double bonds that are health-wise worse than saturated fats. You'll see a lot of advertising now for no trans fats and this and no trans fats, because for a while they were adding those because it was an artificial man-made fat that worked really well in snack foods and preserving things, but it's really bad for you. And so now you'll see a lot of advertisements for not having that. So here is the tail end. This is the fatty acid here. And here's your glycerol. And this just shows you a fat molecule will have several of those. Saturated fats, again, solid at room temperature, very straight, no double bonds. Unsaturated fats are going to have this double bond right here, not right there, double bond right here, which will actually put a bend in it. And that means that if you're packing them closely together, they can't get as tightly packed, and so they are not going to be solid. They're going to be liquid at room temperature. Lipids are also important in the cell membrane. Phospholipids make up the cell membrane. That means a phosphate group and some lipids. It shows well how form fits function. We'll show a little bit about that in a minute. It has two parts, heads and tails. The heads are hydrophilic and the tails are hydrophobic. Hydrophilic, hydro meaning water, philic meaning loving, so they actually are comfortable being around water. And then the hydrophilic ends are water-fearing, so they actually do not want to be around the water environment. The cell membrane is designed so that the hydrophobic parts are away from the environment because they don't want to be near water, and the hydrophilic portion is in contact with the environment, which more than likely has some water to it. Steroids are lipids with 4 fused rings. You may not know this, but a cholesterol is actually a steroid. It's part of the cell membrane, but also can be too high and contribute to atherosclerosis, so heart disease. And then your hormones, estrogen, testosterone, those are all steroids as well. This shows a phospholipid. This is the structural formula. You'll see this is where it gets its name, phospholipid. It has a phosphate. And then here's that fatty acid tail. And this is just a more generalized what we draw when we talk about the phospholipid membrane. We don't draw this fancy stuff. And the way that a cell membrane is made is that the cell membrane, although it looks like a single layer, it's actually 2 layers of these phospholipids. And you'll notice that the tails are in here away from the water environment because they are hydrophobic. And then these heads are going to be hydrophilic, so they're comfortable. So the inside of the cell is going to have the heads, and then the outside of the cell is going to have the heads. And then there's going to be a layer of those tails on the inside. And this is a steroid, the structure of a steroid. I think that's cholesterol. All right, and that's it for this particular video. We just did carbohydrates and lipids. And the next one, we're going to look at proteins and nucleic acids.